The overall goal of this procedure is to image biomolecules such as DNA and proteins in aqueous buffers in real time using atomic force microscopy. This is accomplished by first preparing biomolecules to be imaged. Next, the atomic force microscope is set up and the AFM cantilever probe is positioned. The sample surface and biomolecules of interest are then imaged and analyzed. Ultimately, results can be obtained that show general size, amount, and organization of DNA and proteins and heterogeneous biomolecular complexes in buffered conditions through atomic force microscopy.
This method can help answer key questions in the molecular chaperone field, such as what is the stoichiometry of chaperones and co-chaperones complex to a client protein.
Visual demonstration of this method is beneficial, as the AFM and cantilever probe preparation steps may be difficult to learn from printed instructions, largely because of the intricacies associated with their physical manipulation.
Demonstrating the procedure will be Morgan, Shannon, and John Goertz, 2 students from my laboratory.
To begin, isolate the protein or DNA molecules to be imaged and suspend them in an aqueous buffer. After confirming the purity of the sample, incubate it in an AFM adsorption buffer on ice to promote adherence of the sample to mica. For protein samples, use a 10 millimolar Heaps buffer, and for DNA, a magnesium-containing buffer composed of 10 millimolar tris, pH 7.5, 10 millimolar sodium chloride, 2 millimolar magnesium chloride is suitable. To mount the AFM probe, place the liquid cell probe holder onto the corresponding cantilever installation docking station. Locate the long, thick cantilever of the sharp nitride lever probe to be used.